Longevity Futures. I'm your host, Chris Cohen, and today I'm delighted to be talking to Professor Leslie Lyons. Professor Lyons is a professor of comparative medicine at the University of Missouri. He also is the director of the 99 Lives Cat Genome Sequencing Initiative, which is a project that will read the entire DNA sequence of each of the 38 cat chromosomes. The data generated by this undertaking will be used to identify DNA mutations causing interesting traits, such as the different coat colors and ears and tail types, and even their health problems. So, welcome, Professor Lyons, and thank you so much for joining us today.
Thank you very much, Chris. It's wonderful to join you today.
It's really great to have you. And so, the first question might be a bit of an obvious one, but I have to ask it anyway. Are you a cat or a dog?
I have to say I am a cat person. I was raised with cats, and we always had at least one cat running around the house, never any dogs. I certainly know a lot about dogs from being there's a lot of good canine geneticists that we interact with, and we try to share information between species all the time. So cat, dogs, horses, they're all companion animals, and we try to learn from one another all the time, but definitely more a cat person. Cats.
Definitely not surprised by that answer, but had to ask it anyway. But yeah, can you tell us a little bit more about your background and how you actually got interested in the field of feline genomics?
Yeah, actually, so cats kind of picked me in the end. I was trained at the University of Pittsburgh in Pittsburgh, Pennsylvania, in human genetics. And so it was during that time that I was learning about human genetics that I saw that many groups were doing the same thing and being highly competitive. And I thought, what could be a different niche that I could do that would be very helpful to human medicine, but perhaps a bit different? And I started to realize other people did the exact same things that you do in humans, but it could be cows or dogs or horses or some other species. So I started investigating and talking with investigators that were working on different species, and that was known as comparative genetics at the time, and somewhat still is. It's very similar to comparative medicine, comparative genetics. And I considered working with cattle and fish and cats. And after I got my PhD, I ended up working with cats because I joined the lab of viral carcinogenesis at the National Cancer Institute in Frederick, Maryland. And they were still doing a lot of human genetics, which I wanted to maintain some of those ties. I wanted to learn more about evolution, and their organism that they were studying happened to be the cat. And then also my skills were in disease gene mapping and trait mapping. And that was something that needed to be added to that laboratory as far as the cat work. And so it all fit together. I didn't really choose that because of cats, but I chose the career, and cats were the model organism that were being used. But actually, since then, usually your postdoc sets what your career is going to be. And since then, we haven't looked back. It's been all cats.
Amazing. I think cats are very pleased that you're in their sort of field. Has it ever been
Yep.
tempting to jump back across to humans, or once you're with cats, you thought, right, let's stay here?
Not really, because I've been able to work with many different species. So we have publications on a variety of different species. I work closely with the horse community and also with the dog community here at University of Missouri. And the things that we do in cats relate back to humans. And so in effect, I'm actually do work on humans. The whole idea is the cat is a biomedical model for human disease for certain diseases. And so I do hope that the things that we discover in cats are then applied to human medicine as well. So I'm not really out of human medicine, but I attack it from a different approach.
Well, I think that's a really interesting story, and I think we'll come back to that definitely because it's definitely something I want to touch on, the comparisons between the species really and the model organisms that we use to look at humans. But a bit more about the 99 Lives Cat Genome Sequencing Initiative. Could you just tell us a little bit more about what that is and what are your goals and the aims of the project?
Yeah, the 99 Lives Cat Genome Initiative or project, whatever you would like to call it, is really an effort that I started to push along once I got here to the University of Missouri in 2013. At that time, also, the cost of sequencing was drastically dropping for humans and then also for other species. So a key thing that happened was Illumina came out with an instrument called the X10, and that allowed sequencing costs to dramatically drop to about $5,000 to $3,000 per mammalian genome. So mammalian genomes have 2.4 to 3 gigabases of DNA. And to be able to sequence those genomes at a high enough depth of coverage, that cost had to get down to about that. And so then we realized, hey, it's at a cost that the normal person and researchers can really start sequencing cats. And 9 cats didn't seem like enough, 9 lives. So we made it, well, let's set a goal that we can, is a little ambitious. Let's do 99 cats. And so there's the 100K project and the 10K project for other species. And so with cats, we just changed it up a little bit. And so I've just pushed that forward. And in the end, we have now 30 different institutions, 50 different collaborators from all around the world contributing genomic sequences to the 99 Lives Cat Genome Project.
And how far through are you with sequencing those 99, the goal of 99 cats? Are you close to it, or have you already done it?
We hit that years ago.
It's
So
just
actually,
falling through
yeah.
the data, I see.
Yeah, so all the genomes have been sequenced, and we try to keep high what's called depth of coverage for these genomes so that we know we have very accurate data. So we reached that goal within a year or so. And now we have more than 400 cat genomes. And in fact, we just did a doodle poll. And so the whole consortium is going to get together and regroup and say, okay, what's our next goal? What should we do now? Should we start forming little subgroups that are working on specific diseases such as diabetes or FIP or amyloidosis, hypertrophic cardiomyopathy? So we know those are questions that are out there for cats. And can we do a better job at the consortium? People all kind of work together. And so we'll be asking some of those questions at a consortium meeting a little bit later here in probably September, October.
Some of the results that the initiative has already thrown up, have there been game-changing findings in the field of cat genomics that just have taught us so much that we didn't know before?
One of the biggest things is we can find things faster. And just if you're in humans and you're doing whole genome sequencing to try to figure out why a young child might have a health problem, doing whole genome sequencing, you have about a 50-50 chance of finding the mutation that you think is going to be causal right off the bat and really rapidly. And so that's what we've been able to do with cats. So I think there's at least a dozen papers now that have the 99 Lives Consortium in the author line of different diseases that have been identified, and working on a host of them right now. And we find traits as well. So most recently, mutations for what's called the ticked pattern in cats. A cat that looks like an Abyssinian is ticked. It has no tabby stripes on it. And so my group and another group, Dr. Greg Barsh's group with Chris Kalin, has also done excellent work on the ticked locus. And so we find fun things, coat colors, which in the end are very interesting from the point of biology of how patterning works in animals. But then also we find inherited diseases. And the goal is to do precision medicine. You send us DNA from your sick cat, we find the mutation, and maybe once we know the DNA mutation, you can target a therapy towards that mutation that would be a better treatment than just treating symptoms. And so we hear a lot about this in oncology, where they do targeted therapies and targeted vaccines for certain receptors. Well, we want to be able to do that for most any disease, whether it's in humans or whether it's in cats.
So the field of genomics in humans have obviously helped elucidate the pathways and the sort of genetic underpinnings of diseases such as sickle cell anemia and many different types of sort of childhood cancers. What are the kind of diseases that stand out that are particularly common in cats where compared to other species? And what diseases are then actually particularly rare when in comparison? And why are these?
Yeah. Now, understanding why, we probably don't have good answers to that. And maybe that's where we want to get to. So something, for example, that is more, let's say, rare is when cats normally die of old age, they're generally dying of renal failure. So that's very common, or a type of cancer called lymphoma. So you think, oh, cancers are common in cats, but actually not as common as in dogs. So, yes, cats die of old age associated lymphosarcoma, generally intestinal lymphosarcoma, but they don't have a lot of the other cancers that we find in humans and dogs. So dogs are more of a model for cancers, where cats are actually more of a model for, let's say, like infectious diseases. Cats have coronaviruses, and it was because of cats that we did know, not just because of them, but they certainly helped to understand that the nucleoside analogs, remdesivir, actually work and fight off infections of coronavirus. So we knew that a couple years before humans even saw COVID, we were using GS analogs to fight off feline infectious peritonitis.
That's
And
fascinating.
yeah, and it actually cures it. So that drug actually has provided a cure for what was a 100% lethal disease. If a cat got FIP, it was done for. Other diseases, polycystic kidney disease is a very common human disease. You don't hear much about it, but it's more common than Duchenne, sickle cell combined. And so a lot of older people succumb to renal failure because of PKD, and cats have that as well. And that is something we're actively studying. Can we change the diets, put cats on a more ketogenic diet, and will that help reduce cyst volumes? And we're finding that's true. And the thing is, cats naturally should be on a ketogenic diet, right? So only since the 50s or so have we been putting a lot of carbohydrates in their diets. But now we're seeing that it might be as simple as changing a diet, and that might be the best therapeutic for polycystic kidney disease. Cats have lots of hypertrophic cardiomyopathy. These are the type of cardiac diseases that when you hear about an athlete that suddenly succumbs, drops over from a heart attack, these are hypertrophic cardiomyopathies. And so cats have lots of that. Cats have lots of diabetes. So just like us, they're eating too many carbs, sitting around in the house, not getting enough exercise, and becoming obese. And then they have obesity associated diabetes as well. What we're trying to promote is that now that we can do genomics for most any species, let's pick the right animal model for the right disease. So the mouse is wonderful. We've come a long way with mice, but we need to realize that they're not always the best answer to everything. So they're not particularly the best thing to study polycystic kidney disease, for example. But a lot of research has been done on mice and PKD.
It's the same with Alzheimer's as well, isn't it?
That's right. Yeah, exactly. So let's start learning, because now we have these genomic tools, these whole genome sequences of most any species now. Let's use the right model for the right disease, become more efficient with our science, and get to our therapeutics.
What is the cat model the right disease to model? Or what is the cat the right disease to?
Cats are absolutely, for example, the right model for polycystic kidney disease. A cat's kidney is large enough that you can do a CT or an MRI and not only measure the kidney volume, but you can determine the cyst volume as well, which that's really hard to do with a teeny little mouse kidney. And the thing is, to figure out if your therapeutic is working, you want to see the cyst volume go down as well as the kidney volume. So you can't do that with imaging with the mouse, but you can do that with the cat. So the cat is a slightly better model for polycystic kidney disease. And it all depends on, other diseases and what specialty that you're working on, that the cat might be better, or maybe the dog might be better. Type 2 diabetes, the cat is a pretty good example.
I want to rewind a little bit back to when we were talking about cancer, because I thought that was really fascinating what you said, that cats, there's a one type of cancer which is very prominent, but other types of cancer cats aren't as prone to them. And I know you said the answer as to why these are a much harder question, and we might not be able to answer that. But I do want to ask you, why is that the case? Why are cats less prone to multiple different types of cancer? Because we know we see it in other animals,
Hmm.
such as whales and I think elephants, and it is mainly due to their copy variant number, where
Hmm.
they have high numbers of tumor suppressor genes such as P53. So I was just
Right.
wondering, is that similar to cats, or is it for a different?
We haven't really investigated that well yet in cats. And the two things we have to realize is there might be an ascertainment bias in cats. So, like dogs are always thought, oh, they have all these cancers. There's far more breeds of dogs. So there's more inbred populations, and each one of those inbred populations will have their own health concerns. And they tend to be a lot of different cancers for dogs. So there's more breeds. There's also more vigilance. More of our dogs are fancy breed dogs, where most of our cats are just random bred cats. They're not of an inbred variety. And so we say dogs have more cancer, but maybe that's just also partly an ascertainment bias. And then the second thing also is that, remember I mentioned that we have a 50-50 chance of finding our disease mutations. That's because maybe a good proportion of disease mutations are in the coding part of genes. That's what we are good at. But we're discovering that the ones that we can't find are potentially copy number variants or structural variants that are outside of the genes. We used to call that the junk DNA. Now we're discovering that's important DNA, and now they're calling it the dark matter.
Mhm.
And now we're discovering that the 3D structure that the DNA makes as it folds is actually part of the regulation of genes. And that is what turns genes on and off to some extent as well. So it's becoming very important to look outside of the genes into that dark matter. And maybe that helps explain some of our cancer issues in dogs and other species as well. So, why cats don't have cancers as many? Potentially just an ascertainment bias, but potentially maybe their genome is slightly different. And wouldn't it be cool to find out why they don't have cancers as opposed to why many animals may have a cancer, such as humans and dogs? So the reason why they don't is just as important as the reason why they do.
The dark matter of the genome. It sounds so mysterious, starting
Yeah,
to sound
yeah.
like a physicist there. I love it. It's great.
People like to put
Um,
sexy names on it. Yeah, yeah.
Now, another part of the sort of genome, which is another non-coding, is the telomeres, and
Mhm.
it has become a part of the genome which has gathered a lot of interest in the longevity field. Not so much now, but maybe 5, 10 years ago. But although there's still a lot going on around them. So,
Mhm.
what are the dynamics of the cat telomeres? Because in mice, the telomeres are much longer when they're born than in humans, but then they actually degrade far quicker. So, what are the dynamics of the cat telomeres?
We're just getting to the point where we can answer that. So we haven't, the mouse, second to human, the mouse has a very good genome. And so their telomere structures, how long each telomere is, and telomeres are the caps of the chromosomes. And they pretty much tell the chromosome, okay, you're running into this repeat sequence and there's not going to be any genes there. This is the end. And but now we're discovering that maybe some of that repeat sequence wears away, and when it wears away, does it actually wear away into the next coming gene, and does that affect some of our aging process? So the thing is, we don't know that yet for cats. We know they do have telomere repeats. But now, with the new ultra long contiguous cat genome reference assembly that is now coming out, so we're going to change the reference assembly for the domestic cat. Cinnamon the Abyssinian is going to be retired. And this fall, a new cat is coming in. The cat's name is not as nice. It's Grumpy the cat, and so maybe we'll come up with some pseudonym for Grumpy, but pseudonym. But we have this wonderful sequence that goes telomere to telomere. And Bill Murphy's group at Texas A&M is working hard on making sure that sequence is all put together well. And one of my questions to him was, do we read into the telomere of each cat chromosome, and how long? How long are some of the centromeres as well? Those are repeat sequences as well. So soon we'll be able to answer that more on a different cat basis and monitor that as cats get older. But first we need to know baseline is, because Grumpy the cat wasn't a neonate. It was a cat that was several years old. So we're going to have a genome assembly that represents a cat that is a few years old. And then we'll be able to go from there. Do we see longer ones in younger cats? Do we see shorter ones in older cats? And now we're setting up the tools so that we can start looking at things like that.
Now, I imagine most of these cats that you're sequencing are domesticated cats. Is that correct, or is it cats of all sort of variations?
Unfortunately, but fortunately, we have included many of the wild felids. So I'll refer to cats as our random bred domestic cats, our fancy breed cats. Then there is a species called wild cat. Wild felids that are in these survival programs can also get inbred and have health problems. So we already solved an inherited blindness in black-footed cats. We're working with polycystic kidney disease in Pallas cats. Lions have a vitamin D deficiency issue that causes their skull to malform as they grow as cubs. Fishing cats have transitional cell carcinomas. Black-footed cats, again, also have amyloidosis, and snow leopards have a multiple ocular defect that we're also working on. So not only are we working on domestic cats, the normal average alley cat, Moggy, but fancy breed cats, but also wild felids as well.
So I was in Morocco recently, in Marrakesh, off on a little vacation of my own, and there are a lot of not domesticated, but not wild. They're stray cats, but like you said, they're probably. And it got me thinking, if you compare the genomes of domestic and more wild cats, what parts of the genome have been altered in response to actually living with humans? Have you looked at these sort of evolutionary changes in the cat species, or is that still something that you need to actually have a look at?
Yeah, we would love to do that project, actually, but it's rather hard to do because the progenitor for the domestic cat, which is the African wildcat, Felis libica, still exists. And we can go out and get samples of African wildcats, and we would be able to compare them to domestic cat. And there's also European wildcats as well, but they are not the exact progenitor. They're a step away. They're a different subspecies of wildcat. But the problem is, if you go out and get a sample of those cats today, they've been what we call introgressed. They've already bred with domestic cats accidentally. And so it's really hard to get a real nice clean signal. So that's where we have to go back and try to do ancient DNA studies. And there's a couple groups, one group there at Oxford and one group in France that is very actively looking at ancient DNA. But it's hard to get ancient enough because we need to get ancient DNA that would be greater than 8,000 years ago, because that's about when cats became domesticated. And we want a signal that has no integration. There's no mix of the domestic and the wildcat. We are working to do some things like that. We have in the last genome paper, there was some information on neurological genes that seem to be common in the domestic cats, but that's all very large clouds of data instead of pinpointing, saying exactly here's the gene that causes cats to be domesticated. That's what everybody would love to have. And who knows, there's probably more than one. Cats were probably domesticated differently in the Far East as they were in the Mideast or in different parts of the world. So you never know if you just have only one gene or different parts of the world. There might be different genes that are selected for domestication.
Yeah, I can hear it from your voice. How much of a sort of interesting topic this would be to delve into, the genealogy and the evolution of domestication.
That's probably one of the goals, the next goals for 99 Lives. Can we get more focused on that aspect? But finding those clean data sets is that we can get wildcat samples, but they're modern wildcat samples. And then you're never sure whether they're going to give you the right signal or not, because maybe 2 generations ago, those cats interbred with just feral domestic cats and stuff. But we've done a lot of looking at feral cats around the world, and we know that at the fringes, the cats are genetically different. So Southeast Asian cats are very different from Western European cats. And just like you saw in Morocco, that's how we've collected some of our cats. Just go and offer the cats of the streets in the Medina, just offer them some tuna fish and they come running.
Yeah,
And
my girlfriend did the same. I can definitely attest to that. Yeah, they're all happy with a bit of
yeah,
free food, that's for sure. But yeah, what does the difference between when we have domesticated cats who have been living in the home, does that really change the lifespan and the longevity of the cats we see? If you compare the average lifespan of a domesticated cat to a feral cat, is there quite a difference there because the domestic cat has been looked after in a home? Or do we see a greater abundance of things like diabetes from not being fed right and different sort of health problems? Yeah, I was just wondering, what is the difference in lifespan between these two different categories of?
that's a little hard to put a number because that would mean someone has to
Mhm.
be watching a group of cats for quite a long time to see what did they die of and how long did they, when were they really bold, are they, and that's a little tricky to do. But certainly if a cat is in a household, it's going to have far more protection. And certainly cats, at least in the United States, if they're feral cats, they buy prey, coyotes, by dogs. I mentioned two reasons for cats to lose their lives. That's renal failure and lymphocytes. One of the other major is being hit by a car. That's if you're a household cat and not going out much, then you at least have a better chance at living longer. And my cats have generally lived about 14 years and have succumbed to renal failure each time. But certainly I would think with better protection, nutrition, uh, more consistent nutrition. I don't want to say it's necessarily better because a cat might be out there eating bunny rabbits and mice and lizards and bugs. By far might be the better nutrition for a cat, but having a consistent diet, we do know our pet foods are nutritionally sound. We should be living long in our house. True, to go into the literature and see how well we can document that. But I've definitely seen many cats make it into their early 20s. Yeah.
Oh yeah, I had a cat quite a while ago, our family, that used to eat dinner at our house and then go round to the neighbours and eat a second dinner there, which
Yeah. Yeah.
definitely wasn't ideal nutrition for them. But it's so hard to keep. We spoke to Matt Cabling recently, who heads up the Dog Aging Project in
Yeah.
Washington. And one of
Yeah.
I don't know if I'm sure you know him, but one of
The golden retriever
the sort
study.
of
Yeah.
benefits.
Yeah.
Exactly. Yeah. One of the benefits of looking at dogs as an aging model is the fact that they share so much of their environment with humans. Dogs barely leave our sides. Is this something, when you're looking at or comparing your findings in cats to then being able to extrapolate them and tell us about humans, is that something you can do as well because they are both companion animals? Even though I know cats maybe have a bit more of a independent sort of personalities than dogs, and they go off by themselves. But is that still something?
Yes, certainly. These are things even when we're doing the sequencing projects, these are the things we have to ask about every cat. What is their household environment? Are we for sure looking for something that is genetic or something that is somewhat environmental? So what kind of exposures are they getting? Even what kind of stresses they get? Cats get a lot more stressed than we think they do. And so it's very difficult to actually go away for a week and put your cat in a kennel where that's easier to do for a dog. The cat needs to stay at the house and people come in and feed them. There hasn't been a lot of longevity studies yet accomplished in cats, but I think we're moving to the point of where now we can, when we see the successes of the Golden Retriever project, and now that we can do web-based tools and web-based interactions with the lay public. So something we would call citizen science. Really, we just need to start launching things like that. Can we start understanding what is common about cats that are living to be 16 years of age and longer, as opposed to what's probably about the normal lifespan of 12 to 16 years for domestic cats? Those type of studies should be able to be launched.
And when we're talking about the translation of sort of the findings in cats to humans. How much of our genome do we actually share with cats? And can cats' genome actually tell us a lot about our own genetic makeup? And what are the sort of major findings that you have found in cats that you believe could actually be extrapolated to humans and be useful in humans?
Certainly, if we look at the coding part of the genome, but however, the coding part of the genome, the part that the DNA is translated into actual proteins, so the actual genes, that's actually a very small part of any mammal's genome, so less than 10%. So when we look at genes, some genes between a cat and a person can be 100% exactly alike, where others can have a lower percentage of similarity. On average, we're probably looking at about 85% similarity between a cat and a human, and dogs about the same. Yes, and mice are actually even a little less as compared to humans. Perhaps the cat is the right model to help us to understand structural variation and regulatory variation that is outside of the genes and makes up 90% of the genome, and will help us understand diseases better in that fashion.
Amazing, amazing. And as one of the final questions, could you say 2 or 3 things that you believe are inevitable for the field of pet longevity over the course of the next sort of 5 or 10 years?
Inevitable is one that I certainly want to make sure does happen, is cats will be part of precision medicine. So we're trying to do that now, that if your pet has a health problem, it can come in, get a genome sequence done, we find the cause for your cat's problem, and it gets the right therapeutics so that you can have better longevity and a better quality and quantity of life for that individual animal. So that's the immediate goal, and that's what I hope is occurring within my lifetime as well. But there's also things we're going to discover more things about cat nutrition that will help us keep our cats healthy and strong, and maybe try to learn out what's going on with renal failure, perhaps get cats to live longer in that regard as well. And then hopefully we'll also understand what's going on with the overall biology of longevity. What genes are involved? Is it all about telomeres, or are there other genes as well? And what kind of nutritional should a cat be drinking a glass of wine every night, and that's why they live longer, or something like that? So those are more speculative, but definitely the precision medicine aspect.
A glass of catnip every night. Yeah, I think they
Yeah,
can get on board
exactly.
with that. And I think
Yeah.
a lot of what you're saying has been, we've hosted a number of talks with people working in human longevity, and I think it seems like the next wave of research is going to be in these companion animals. And once we can show that we can have these really positive effects on the health span and the lifespan of cats, dogs, all of those, that we can actually translate that biology across the humans. So I think it's going to be a really interesting time.
Right.
And
Well,
yeah,
yeah, let's not forget just the behavioral aspects as well.
of
We've
course.
certainly seen that just having pets, their longevity and their quality of life as well. And so does that go both ways? Does a good human companion for a pet, for an animal, help them with their longevity also? When a cat lays on our lap, does it lower its blood pressure like they lower our blood pressure? It's something we have to investigate.
With some cats, maybe, other cats, not so much. Thank you so much for spending the time with chatting to us. Where can people keep up
Yeah,
to date with your work and what you're doing?
all of our work is always published, and it's always open access. So if you, through Google Scholar, you can always find our published work. Periodically, we do from the press, and so you can find us in the press on it. But then also our website at Feline Genome at missouri.edu is where we have our laboratory website as well, and we'll start trying to post better for about the 99 lives as well. And maybe someday we'll be more active on Facebook and Twitter and Instagram as well. But we do have a Facebook account, but it's hard keeping up with all those social media aspects.
No, I completely
Would
agree.
like a volunteer, we'll take it.
I'm all for doing the same. I'm trying to get better at it. But yeah,
Yeah.
please go and check out Leslie's work. Thank you so much for joining me again. It's been an absolute pleasure chatting to you, and it's just incredible work what you're doing. And I can't wait to hear more about it, and hopefully have another talk like this in the future.
Yeah, my pleasure. And remember,
Brilliant.
the cat world is a group of investigators, so it's my work. It's a whole group of investigators from around the world. Veterinary.
Perfect. I think I lost you there for a second, but again, thank you so much. And guys, if you've enjoyed this, please head over to the Gowing Life website, gowinglife.com, where you can find all of our other news and articles, keeping you up to date with the news in longevity. So thanks again for listening, and I'll see you again next time. Thank you very much. Goodbye.
Take care, everyone.